So let's start. Welcome back to this new edition of doctoral meetings. Today we have the pleasure to count on Lara, María, Vázquez, González, who is going to talk about her thesis under the title Bioinformatics Procedures for the Advancement of the Implementation of Clinical Metagenomics. So, yeah, Lara, you can start. In principle, you have like 20 minutes for the presentation, and then a maximum other 20 minutes for discussion and questions. Although I think today we may finish earlier because we are not that many. All right, so go on.
Thank you. Good morning, everyone. Thank you for attending my doctoral meeting. Today I will give an overview of different bioinformatics procedures for the advancement of the implementation of clinical metagenomics. The main objective of my research is the creation of bioinformatics tools to achieve a better analysis and easier processing of the genetic material contained in samples, specifically samples containing microbial DNA, which is the DNA found in microorganisms such as bacteria or archaea. These tools were created in collaboration with the Oral Sciences Research Group from the Faculty of Medicine and Dentistry. And we usually publish the clinical point of view and also the technical approach to create these tools. I'll start with an introduction to several concepts related to metagenomics. The first concept is metagenomics itself. Metagenomics is the study of the genetic material like DNA or RNA found in samples. These samples can have diverse origins, such as food, environmental, or patients. If the samples are from patients, it is clinical metagenomics. We will focus on the study of bacteria and archaea in samples from patients. And to do that, we will use samples obtained through 16S rRNA gene sequencing. The 16S rRNA gene is a gene that is found in all bacteria and archaea, and it is considered a phylogenetic marker, meaning that we can use it to taxonomically identify both of these microorganisms. To do that, we have variable regions and conserved regions. In green, we can see the conserved regions, and in gray, the 9 variable regions. The conserved regions are highly similar for all bacteria and archaea, while the variable regions are different across different species. This gene also can appear usually more than once inside the genome of a bacteria or an archaea, and it can have several copies that can be identical or slightly different inside these genomes. So, how can we find this gene inside the genomes of bacteria and archaea? To do that, we can use primers. Primers are short sequences of DNA that provide a starting point for DNA amplification. They usually have between 18 base pairs and 24 base pairs. Here we have an example of DNA amplification using 2 primers. One forward primer that matches the sequence in one conserved region, and a reverse primer that matches another conserved region. With them, we can amplify the fragment in between that corresponds to a variable region that we can use to differentiate these microorganisms. The primers usually contain degenerate bases. Degenerate bases are bases that can correspond to multiple nucleobases. The nucleobases are A, C, G, and T. And here, for example, we have the degenerate base N that can correspond to A, C, G, or T. Now that we have introduced these concepts, we can specify the objectives. The first objective is the development of a bioinformatic procedure to analyze the coverage of primers or primer pairs on any database. The second objective is the implementation of a bioinformatic procedure to evaluate the intragenomic redundancy of the 16S RNA gene. The intragenomic redundancy is this characteristic that we mentioned earlier of having more than one copy inside a genome. Finally, we have a third objective of developing a bioinformatic procedure to classify phenotypes, which in our case will be diseases. We'll start with the tool created in the first objective, which is PrimaryValPy. PrimaryValPy is a Python package to analyze the coverage of any primer or primer pair on any database. First, we need to define what the coverage is. The coverage is the proportion of target sequences in a given dataset that can be amplified by a specific primer or primer pair. And how PrimaryValPy works is that we receive as input the sequences of the primers and also the sequences of the genes or genomes where we want to test these primers. There's a first step for quality control where we check the target sequences, which are the sequences of the genomes and genes under study, for any degenerate bases that can affect the analysis. Then there's a second step that we'll skip for now, and then we can proceed to look for the primer sequences in the target sequences. We do this using fuzzy regular expressions. We use fuzzy regular expressions in specific because we allow for mismatches between the primer sequence and the target sequence. That means that we allow for a number of differences between these sequences. Then the coverage is calculated and the amplified fragments by the primers are returned. The step that we skipped is a step for grouping the target sequences depending on a specific taxonomic level. For example, we may want to group these genomes by species, and then the calculated coverage will be the number of species amplified by the primer instead of the number of genomes amplified by the primer. We used PrimaryValPy to test commonly used primers in the literature against a bacterial and archaea oral database. Aside from obtaining the most suitable primers for detecting archaea and bacterial in the oral cavity, we also saw that many of the primers that were supposed to cover an exclusive domain turned out to cover a different domain than expected. For instance, 356 of the primers were expected to exclusively amplify bacteria, but it turned out that in the oral cavity only 200 amplify exclusively bacteria. The remaining ones ended up amplifying both bacteria and archaea. Furthermore, from these primers that we tested, we saw that 26 had no coverage at all in the oral cavity. So this highlights the importance of using a tool like this prior to any experiment in the laboratory. So we pre-select the most suitable primers for the niche under study. With this work, we have two associated publications, one in BMC Bioinformatics explaining the procedures of PrimaryValPy, and another one in Microbiome explaining the clinical implications of this work. Then we move on to the second objective, which is the creation of the 16S GeoSec database. To explain this, we need to explain a little bit more the concept of intragenomic redundancy. Intragenomic redundancy was the characteristic of the 16S RNA gene, that is that we can have more than one identical or distinct gene per genome. This is important because there are technologies that estimate microbial abundances based on these gene counts. And in these types of technologies, microbes that have fewer genes may be underestimated, while those with more genes might be overestimated. Furthermore, this gene count number can be different depending on the niche. One specific bacteria can have a different gene copy number in the mouth than in the gut. So we propose 16S GeoSec, a database with gene copy numbers specifically for oral bacteria and archaea, that we created from complete genomes of oral bacteria and archaea that have also available their complete taxonomy up to the species level. We obtained them from two databases, the NCBI and the EHOMD. And then we extracted the genes from these genomes using an algorithm called Search16S by Robert Edgar, which is an algorithm that has a sensitivity of over 99% to known 16S genes. We have two publications associated to this work. One is the database itself that is currently under review in Scientific Data, and another is a publication in Microbiology Spectrum exploring the impact of this gene redundancy in next-generation sequencing. Finally, we move on to the last object of my research, which is F-class. First, we need to explain the concept of dysbiosis. We know that some diseases can be caused by specific bacteria, like, for example, Salmonella. But there are other diseases that are actually caused by an imbalance in the microbial communities present in the patient. And these are harder to diagnose because there is no specific bacteria to blame. Examples of these diseases are Crohn's disease or periodontitis. So we propose F-class, which is a curated pipeline for phenotype classification. In this case, this is classification based on 16S RNA gene count tables. To perform this classification, we use ASV abundance tables. ASVs are amplicon sequence variants. We earlier said that in a genome we can find copies of the gene that are identical or different. So the variants of a genome are the copies that are different, removing those that are duplicate. This is an example of an ASV abundance table where we can see for each sample its composition depending on how many ASVs of each type it has. For example, sample 1 contains 155 counts of the ASV1. This data has compositional nature. The first module of this pipeline is the data processing module, where we first filter the abundance table by removing the samples that have a total number of counts really low, and we also remove from the table the ASVs that are less significant, those with less number of counts overall. Then we can proceed to split the data into 70% for training and 30% for testing. And optionally, we move on to a data augmentation step. If the dataset that we're using is imbalanced, we would perform this step. Here we recommend using algorithms that take into account the compositional nature of the data. So the new data points created are still compositional. Then we have to transform the data using centered log ratio transformation, which is especially recommended for compositional data like abundance tables. Finally, we perform a feature selection step to further narrow down the ASVs in our table. We use recursive feature elimination with several estimators like random forest, logistic regression, and k-nearest neighbors. At the end of this module, we have our processed ASV abundance table. Then we can move on to the training and evaluation module, where we evaluate 5 different models: k-nearest neighbors, random forest, support vector machine, XGBoost, and multilayer perceptron. We first perform a hyperparameter tuning step to tune the parameters of the different base models. Then we perform cross-validation, specifically tenfold stratified cross-validation. And then we create ensemble models. To create these ensemble models, we decided to use dynamic ensemble selection techniques that choose the best possible ensemble for each new sample that we're trying to classify. The idea behind this is that not every individual classifier has to be an expert on classifying every unknown sample, but rather each classifier can be an expert in a local region of the feature space. we tested 4 different approaches for dynamic ensemble selection. Finally, we trained all these models and the ensemble models on the training data and evaluated them on the 30% of the test data. Sorry, on the 30% of the data, which is the test set. I will briefly comment the application of this pipeline, which we use to diagnose periodontitis and gingivitis. We used a dataset containing samples from 3 collection sites, samples from saliva, from the subgingival site, and from the supragingival site. We decided to group samples from both subgingival and supragingival sites because they both are located in the plaque. And then we divided this dataset into 4 different subsets, depending on the disease, if we want to study periodontitis alone or periodontitis with gingivitis, and depending on the collection site, plaque or saliva. Just as a note, gingivitis is considered an early stage of periodontitis. And we start with over 10,000 ASVs. Here we have the results, the F1 scores after cross-validation for each of the subsets. We can see that we explore different numbers of features. That is because when performing feature selection, you have to specify the target number of features that you want to reach. And we weren't sure which number of features would work best. So we tested several values from 2 to 15. Here we can see in the solid lines that the ensemble models performed generally better than all individual models. So how can we decide which one is the best ensemble model for detecting this periodontitis and gingivitis? We chose to average the F1 scores throughout all the features. Here we have the average scores for the ensembles. For all of the subsets, the best ensemble was the dynamic ensemble selection clustering, which, for example, for the subset containing samples with periodontitis obtained from saliva, achieved a F1 score of 0.956. Then we tested this ensemble with the test set for the 4 different subsets, and we achieved, using 9 features, an F1 score of 0.921. This work has a publication currently under review submitted to PLOS Computational Biology, where we explain this pipeline. As conclusions for this work, this thesis, we have developed 3 useful bioinformatics procedures. The first, primary val-pi, to analyze the primary coverage on any database that can reduce the number of necessary experiments in the lab. Then we developed the database 16S GeoSec that offers gene count numbers for all bacteria and archaea and can be used in technologies that need to estimate microbial abundances. And finally, we developed FClass, a pipeline for phenotype classification that can be used for harder to diagnose diseases like dysbiosis-associated diseases like Crohn's disease or periodontitis. Thank you for your attention.
All right, I don't know if anybody has any question over there. No? All right, so, well, I see that you have covered already the 3 objectives that you considered in your thesis,
yes.
which means that you are close to,
yes.
to finish, right?
Yes.
Okay, very good. So I have, of course, some questions, because I am probably the most outsider person in the audience. So I have general questions on, for example, the data sets. I was wondering whether they are being taken from existing data sets that are open or are within a research project in which you are working on?
Okay.
How it works, and or someone else picking samples for you?
Yes.
And then they are using your library. So if you can explain a little bit about how is this going on?
So, okay, for example, for the creation of the gene copy number database, we used samples, we used complete genomes of oral bacteria and archaea that are already publicly available in databases like the EHOMD and GenBank and Taxonomy from the NCBI. So these, anyone can access them. For the data used in FClass to diagnose periodontitis and gingivitis, the samples were actually obtained by our research group, those from the Faculty of Medicine and Dentistry.
Okay.
And for primary Valpi, we used a public database containing genes, or 16S RNA genes found in the oral cavity. Those, the ones that were already published were the samples from the bacteria. But for the archaea, we actually had to compose our own database searching for the individual genomes of each archaea. So I don't know if that answers your question.
Yeah, well, I wanted to know, I was curious about whether you are just relying on publicly available data or there is something else going on in the research.
Both depending on the...
Yeah, that's nice that you are collaborating with faculty. Also, I have some additional... I believe that the pipelines, perhaps you may have described a little bit more detail, right? Especially because we are not experts of in your field. So the first question I had is basically, I imagine that much of the work that you did has to do with having like a sort of data structure to look for the patterns. I don't know if this is correct, or so are you using any special data structure for preparing your
There's, for example, in FClass, there are some bioinformatic procedures applied to the samples once they are obtained from the sequencing platform. They are known pipelines that check the quality of the sequences, remove those sequences that don't have enough quality, and that also remove, for example, the primer sequence from the amplified sequence. So all the samples all the sequences that we work with have been under this bioinformatic pipeline. In the case of FClass, we performed it, but when using already published samples, that is already done.
Okay, questions? Okay, so, well, there's people here that I know that they use data augmentation techniques, right, in different domains. So I was wondering whether when you are preparing your classifier for training, which is your last stage in your thesis, can you explain a little bit more about the data augmentation techniques that you are using?
We used...
because in the image domain and in text domain, we may easily think on how to augment this data. But in this domain, I'm not sure how it's done. So if you can tell us a little bit more.
So here, we actually used an algorithm called compositional cut mix, that what it does is when creating new data points, it makes sure that the new samples also follow a compositional nature. So when we add up all the relative frequencies of each ASB, it still adds up to 1. I cannot exactly tell you now how the algorithm works, because we used this recommended one, usually used for compositional data, but that's the principle behind it, to maintain the compositional nature.
Okay, one more thing. No questions yet? How large are the databases that you are handling? I'm curious about it.
So here we used almost 2,000 samples, 180. And
how large are the samples?
So the samples usually contain around 2 megabytes of data, each sample.
How many?
2 megabytes. 2 megabytes. Okay. So we have loads and loads of nucleotides sequenced in these samples. Wow. However, again, in FE class, we work with the number of variants found in those samples. So we have tables of abundance, not the samples itself.
Okay, thanks.
So,
yeah, I don't know. These are the main questions I had. So, I don't know if your supervisor had any comment or any critic that is always useful for you. No? Okay, so let's see some of the notes that I took. Yeah, I think, yeah. Which is the feedback as you are collaborating with other people, along with your supervisor, with the faculty? Well, I have actually two more questions. So one is, have they tested your software, or are they intended to use it? Or is this intended to be integrated in something that they can use easily? Or which is the expectations?
So, for the first objective, which was primary Valpi, we did a study to find the best primers to amplify bacterial archaea in the oral cavity. Ideally, we would test those in the lab to check that they actually are suitable for covering these microorganisms. And in the case of FE class, again, ideally, it would be used as a diagnosis tool to avoid the stress on patients of having several clinical trials that can take longer. So we yet haven't applied them, but we intend to.
Okay, it would be great to in the future, right? Because it would be nice to see the usefulness in practice, right? Yeah. A related question, therefore, is, I saw one of your papers, it's about the library that you implemented.
Yes.
Is this library closed?
It's open source. It is public in GitLab. So.
All right, and then are you aware in other labs? Or?
It was just published a few months ago, so
okay.
Still not.
So you still have to make good publicity of your library. So yeah, all the best. Good luck with that. So congratulations. I think if there's no more questions over here, comments.
Thank you. I want to say that the table we are seeing is very important, because to diagnose the periodontitis, it's very uncomfortable for the patient. But if with a how do you say muestra?
Sample.
With a sample with a simple sample of saliva, you can diagnose these illnesses or this disease. It's very easy to do for in the clinic.
Yeah.
So we are working in this, and we want to extend this to diagnose, for example, oral cancer or other diseases that worry much more to the population, and we are working in this sense.
Yeah, therefore. Right. Okay, so congratulations and thank you for sharing your thesis topic
Thank you.
and your presentation. So an applause and we are done.